Hello, everyone. My name is Nick Orfanos. I act as the ICH coordinator for Health Canada. I will be facilitating the Q&A session for this meeting, along with Jill Adleberg from the US FDA, who you heard from in the ICH overview presentation at the beginning of the meeting. Before we get started, I just wanted to thank all the speakers today for very informative presentations. Today, you heard presentations on guidelines that have recently reached step 2 of the ICH process, which again, as a reminder, is a draft guideline that was recently finalized for regulatory consultation in the ICH regions, as well as guidelines that have recently reached step 4 of the ICH process, which is essentially guidelines that have recently been finalized at ICH and moving towards implementation in the ICH regions. Today, you really heard a subset of the work of a much larger number of topics being undertaken by ICH, and really shows the magnitude and impact of the work of ICH. So without further ado, we can get right into the Q&A, and I will start by asking questions for Jill, who presented the ICH overview at the beginning of this session. So the first question for Jill, you touched on this briefly during your presentation, but how does ICH determine which new topics are chosen? What is the process, and what are the criteria considered in deciding a new topic?
Thanks very much, Nick. That's a great question. So there are really a number of factors. The first is capacity, or the availability of expert resources to take on a new topic. For example, when the pandemic hit, ICH had a really large existing workload of quality topics and a lineup of priority quality topics that were waiting in the wings. During the public health emergency, our experts acquired added responsibilities that had them struggling to balance overstretched workloads. As a result, the ICH Management Committee and Assembly recommended a temporary halt to the consideration of new quality topics to allow parties to catch up. And in the past few years, that's exactly what the working groups have done. Another factor is that there has to be sufficient scientific understanding of a new topic. All parties may agree that a topic is a high priority, but if there isn't sufficient scientific understanding of the underlying issues, the topic is unlikely to be ripe for consideration. And then I'd mention that in recent years, ICH has advanced new topics strategically by developing reflection papers on emerging topics and outlining potential new guidelines or modifications to existing guidelines via the reflection papers. Often the reflection papers lay out a staged approach involving one or more guidelines. For example, the current revision of the E6 guideline was recommended in the Good Clinical Practice Renovation paper, reflection paper, and the General Considerations for Patient Preference Studies guideline was recommended for consideration in the patient-focused drug development reflection paper. So really a combination of a bunch of factors, but a great question.
Great, thank you for that, Jill. So next question, what is the process for offering input and ideas for consideration into draft guidelines when they're released for consultation? And some of those which will probably be relevant for some listeners here today.
So all new ICH guidelines and major revisions to existing guidelines are issued for public comment. Individuals and organizations can provide comments through their country or region's formal public comment process or directly to ICH. If you take a look on ICH's website, you'll see a tab for work products and underneath a link to the public consultations page. Currently, the E2D draft guideline is listed along with instructions for submitting comments directly to ICH. All comments are reviewed and assessed by the expert working group developing the guideline and often lead to changes that are reflected in the final guideline. I might also note that E6R3 developed the first stakeholder engagement plan, which recognizes that patients, academic clinical researchers, and others have unique and important insights on the design and conduct of clinical trials, and their voices are relevant and should be heard. And the E6R3 EWG has regularly sought input from these stakeholders. And I think those are the main ways, Nick. Back to you.
Great, thanks for that, Jill. I think we have time for a couple more questions. So next one, I think this is one that we're all sympathetic to, but what has been the impact of the substantial growth in new ICH members and observers over the years?
So it's really been positive. It's contributed to an infusion of new talent and diverse perspectives. And it's also given smaller countries and regions the opportunity to network and learn from more experienced and well-resourced colleagues who are managing many of the same challenges.
Great, thank you, Jill. And one more question. I understand that the regulatory members of ICH meet separately to discuss ICH issues. Can you explain a little bit more about this?
Yeah, another good question. ICH has been tremendously successful because it includes both global regulators and industry experts working together to develop consensus-based guidelines. Regulators obviously have a unique role and perspective, and they do meet regularly under the rubric of the International Pharmaceutical Regulators Program, or IPRP, to exchange information on issues of mutual interest and to enhance collaboration and regulatory convergence. IPRP members typically meet immediately after ICH biannual meetings to discuss implementation of ICH guidelines and other standards, and to share information on global regulatory issues and emerging technologies.
Great, thanks very much for that, Jill. So Jill, I'll pass it off to you for the next set of questions for the next presenter.
So our next set of questions is for Kellie. Kellie, when DDI studies are designed, how are the drugs that could possibly interact with the therapeutic product of interest selected?
That's a great question, because obviously there are endless numbers of drug combinations that could be studied. But so the guideline does address this question. The initial decision to conduct a clinical drug interaction study is based on in vitro study results. So if the in vitro results indicate that a drug might inhibit or induce an enzyme or transporter, or it's a substrate of an enzyme or transporter, the drugs to include in the study are selected based on that mechanism. So the tables in the appendix can help select the drugs. It's preferable to use an index drug if one's available for the specific mechanism. And this is because the results of an index study can be extrapolated to other drug combinations. However, if an index drug is not available for the mechanism, or if the index study has already been conducted, it's typical to select a drug based on mechanism, so there may be an interaction, and the importance of the other drug to the intended patient population. And there may also be risk considerations. Is it considered a risky combination? Thank you.
That's great. That's great. The next question is, are there significant differences between the draft guideline and what we can expect in the final guideline?
Most of the changes were really for clarity. There were some comments that indicated that there might be a misunderstanding of what was in the draft guidelines. So that's what most of the changes were based on. However, there were some changes, and this includes some topics that I included in my presentation. There's more information about when the protein binding measure doesn't need to be capped for a highly protein bound drug. There's more information about the use of endogenous biomarkers to determine the potential for a drug to affect transporters. And there are also some changes to some of the in vitro cutoff values. But other than that, it's mostly clarity related changes.
Great. What issues has the expert working group struggled with most to develop a harmonized approach?
I think the biggest struggle, and this happened during the initial drafting, and then also when we were responding to comments, was really the level of detail and how prescriptive we wanted the recommendations to be. It was interesting that for some of the topics, when we were looking at the public comments, for the exact same topic, we had requests to be more flexible and also to be more prescriptive. And so we had to think about how prescriptive do we really need to be, and when could we just explain a process but not have cutoff values. So that balance was probably the biggest struggle. But there was quite a bit of discussion also around the protein binding topic. the level of detail regarding the use of predictive modeling, because this still is an emerging area. And then there's another ICH guideline related to modeling in general that's really just being started. And then also how to describe the interpretation of drug interaction results, because that can come across as quite subjective. So we had a lot of discussions on that topic.
Great, thank you. One of our participants asked, can drugs that are not listed in the tables in the appendices of the guideline be used in clinical DDI studies?
Yes, that's a good question. Yes, they can. That is certainly permissible, and it's really expected also. There's a preamble before most of the tables in the appendices, and that indicates how the drugs were selected for that particular table. So based on the criteria in that preamble, other drugs may be selected for the drug interaction studies. And then drugs may also be selected based on likelihood of co-administration, the mechanism, and the risk for the patient population.
Great. And our last question for you right now is from a participant who asked, after a DDI study is completed, what criteria can be used to assess the need for a DDI management strategy?
Thanks for the question. And before I answer that, I'll indicate what a management strategy is. So a management strategy, it can typically be an instruction to not use the drugs together. It could be to change the dose of one or the other drug, or it may be special monitoring that's needed when drugs are combined. So in order to consider the need for this type of strategy, it's important to consider the anticipated change in drug concentrations. So will the concentrations go up? Will they go down? And what is the magnitude? And then based on the knowledge of exposure response and for safety and for efficacy, is there an anticipated consequence of the drug interaction? So that's the most basic way to explain it. And that is outlined in the guideline. Thank you.
Very helpful. Thanks, Kellie. Nick, back to you.
Great, thanks for that, Jill, and Kellie as well. So our next set of questions will be for E2DR1 post-approval safety data management, definitions and standards for management and reporting individual case safety reports. Our presenter was Craig Zinderman. So just questions here. So first question is, are there plans to integrate IDMP into adverse event reporting?
Yeah, so thanks for the question. E2D is really a guideline that's at a higher level, what some might call a foundational guideline. It defines terms, concepts, reporting requirements, and approaches. Guidance on a specific taxonomy like IDMP for product identification would be covered in the E2B guideline. It's really outside the scope of E2D, and it wasn't part of the E2D working group's deliberations. That said, there is an FDA guidance for industry called Identification of Medicinal Products, Implementation and Use, and that's from March 2023. And that guidance describes FDA's position and current thinking on IDMP and the 5 IDMP standards and progress so far with implementing them. And it mentions in that guidance sort of a phased approach where FDA is planning to work with stakeholders to globally implement these standards.
Great, thank you for that, Craig. Next question. The new draft ETD guideline says that a username or a handle isn't enough to confirm the existence of a real patient when cases are submitted via a digital platform such as social media, but that the addition of an identifier such as gender, age, or age category would be enough. In a world where fake profiles are increasingly prevalent on social media sites, could you speak about the expert working group's thought process for verifying the existence of real patients on digital platforms?
Yeah, certainly a timely question. While it was a major purpose of the guideline to come together to address the burden and volume of all of this post-market safety data and post-market reporting from a variety of new sources, including digital platforms or internet, social media sources was one of them. It was really about addressing sort of the volume and how to manage all of these cases and how they should be reported. specifically being able to discern the veracity of information on social media or other internet sites, being able to tell what's real and what's not real, what's fabricated by a bot or generative AI, wasn't really part of the goal of coming together to do this revision. That said, it was a big topic for the working group, particularly since many of our discussions happened during the time of, recently with excitement and expansion of AI, really globally. So we provided the criteria that you mentioned for the username to not in and of itself qualify as for patient identifiability, but something else like gender or age would be required to really know the existence of a real patient. So that was sort of a baseline that we were able to provide as a recommendation in the guideline, but really couldn't find other recommendations that we could give that would help, that would really work to tell what's real and what's not real out there on the internet. That was pretty much what was doable as a recommendation.
Great, thank you for that, Craig. And then one last question, very simply put, what's new and different about the revised guideline?
So I addressed a lot of the differences in my talk and focused on them, but I'll pick two that I think are really important to point out. One of them is that the previous guideline, the original guideline, is really about expedited reports only, whereas this guideline really expands the scope to all ICSRs. Part of the reason for that is the original guideline. I think it really directed expedited cases because there was harmonization with the requirements across regions about what was required, what the time frame was, what an expedited report, what was considered to be an expedited report, serious and unexpected. But now that has changed, and there's not as much harmonization, even among what's considered an expedited report. So there wasn't really an advantage to just restricting the guideline to expedited reports. And the concepts that we talk about, digital platforms, patient support programs, definition of an AE, serious AE, really apply to all ICSRs, not just expedited. So we really wanted to broaden it. And the other thing I'll point out is all the changes relative to these new or more frequently used data sources. So for patient support programs, for digital platforms, there are definitions for these things that are provided. These are new definitions, particularly for patient support programs. The definition really requires that the program has two-way interaction, so that considering adverse events from those programs to be solicited really means that those adverse events are derived in the context of that interaction between the company and a healthcare provider or a patient.
Great, thank you very much for those responses, Craig. So Jill, back to you for the next presenter.
Terrific, thanks very much, Nick. So Dr. Legare is unable to join us this afternoon. In her place, we're delighted to welcome Dr. Keire Alzarad. Keire is the E6R3 rapporteur and has expertly led the working group during development of the revised principles guideline, Annex 1 and now Annex 2. In his spare time, Keire serves as the director of CDER's Office of Medical Policy, where he leads the development, coordination, and implementation of medical policy programs and strategic initiatives. So Keire, thank you very much for joining us. Our first question is, why doesn't ICH E6R3 apply to non-interventional trials?
Thanks so much, Jill. Do you hear me?
Yes.
Oh, okay, good. Perfect. And thank you again so much, and I'm delighted to be here with all of you. It's a great question. Actually, the EWG, the expert working group, from the very beginning discussed the inclusion of non-interventional trials, what's called actually non-interventional studies. It's important to note that having an intervention is really a hallmark of clinical trials. Obviously, non-interventional studies, or as a term called like observational studies, do not have an intervention. And if you think of E6 as really an outline of how to manage the risks, but also how to manage and ensure the quality of the data, the reliability of the results. In the context of having an intervention, that's very different than when you would not have an intervention, for example, observational studies. For observational studies, the risk profile in general is more focused on data privacy and confidentiality because of the lack of the intervention. So we thought that's really a very different topic to address. Not that's not important, it is important, but for E6, we want the focus to be on interventional studies, hence clinical trials, and that will give us an ability to provide more of a concise and targeted and reasonable approach across the board, across the different designs of clinical trials. I hope that's helpful.
That's very helpful. Thank you, Keire. Our next question is from a participant who said, I may have missed this, but is ICH E6R3 currently in force? If not, what's the anticipated timeline for implementation?
Yeah, thank you so much for the question. We are on time to hopefully finalize R3 by the end of the year. Right now, R2 is finalized, and it's important to note that there is no contradictions between R2 and R3. R3, we've seen it as more of a modernization step that took the concepts in R2 and really put them in a more of a modernized context, if you would, with proportionality and risk-based approaches at the core of all of that. I hope that's helpful.
Great, thank you, Keire. Next question is, the E6R3 principles are very broad. Do you plan to develop training materials, including examples, to help sponsors better understand some of the concepts discussed?
Yes, great question and very timely one. As we developed E6, we realized that there is a lot of areas that worthy of a little bit deeper dive, if you would. And we made a list. This list now is a couple of years old and it keeps accumulating. We made a list of the key aspects that we would like to address in the training. We're hoping this training will not just be a repetition of what's in the guidance, but it will have use cases, for example, novel trial designs, trials with decentralized settings, trials in healthcare, embedded in healthcare, and to show through that training itself how GCP can be flexible and agile in different contexts here. So yes, the training is a primary aspect of what we would like to do, and we're putting quite a bit of onuses and hope on the training to complement the guideline itself.
That's great. The next question is, were all public responses and comments provided on E6R3 reviewed? How were the numerous comments taken into consideration for updates? And finally, were the comments reviewed during the series of joint meetings?
Yeah, it's a good question. We've received around 6,000 comments during the public consultation period. We were really excited to see the level of interest in E6. We are actually very much committed to really looking at every single comment. What we did, each agency looked at the comments that we received at the specific regions. Like for example, for FDA, we looked at everything collected from the US. And what we had to do is to put those comments into themes. The group came up with, I think, approximately 17 or 18 themes that the totality of the comments really fits within those themes, with a few exceptions. Obviously, we have a miscellaneous category. And now the group, through their meetings, they're going, with on each theme and addressing each element here. It's been really helpful for us to see where the concentration of the comments really try to point us to. And we believe in general, this is going to make the guideline far more robust.
That's great, Keire. So in the interest of giving every presenter an opportunity to answer questions that we've received, let's move on now to go back to Nick and move to M14. And if time permits, we'll come back for a second round of questions. Thanks very much, Keire.
Thank you.
All right, thanks everyone. So yes, our next presenter was on M14, which is General Principles on Plan, Design, and Analysis of Pharmacoepidemiological Studies that utilize real-world data for the safety assessment of medicine. So Ken from Health Canada was our presenter here. So one question for Melissa. A reflection paper on harmonization of real-world evidence terminology and convergence was released last June for public consultation. How does the topic of that paper relate to or complement this M14 guideline?
Thanks so much for the question, Nick. And it's a really good question. So the draft reflection paper, and it's available on the ICH website for those of you who may not have had a chance to look at it during the public consultation last year. It's really meant to address some of the broader challenges that we have related to harmonization and convergence that we have both in the real world evidence and real world data spaces, with more of a focus on effectiveness, whereas M14 is more specifically targeted to the safety assessment of medicines. So just as a bit of history for the reflection paper, it really came out of themes that were identified by the International Coalition of Medicines Regulatory Authorities, RWE workshop participants back in June of 2022. So this led to a resultant ICMA statement on international collaboration in the RWED spaces that was released in June of that year. So these were really kind of the ideas of a group of collective regulators trying to address this. So I will say that really in the end, both are meant to complement each other. So both the topic of the reflection paper as well as M14, in the sense that they both focus on promoting the use of high quality data and evidence, and better enabling the integration of both of these into regulatory decision making. So as we have worked on both M14 and the draft reflection paper, we have, of course, been very mindful of avoiding duplication and inconsistencies amongst the two. So, for the reflection paper, it's really outlining objectives, promoting more of the common understanding of, different types and scopes of real world data and evidence, description, ultimately really the goal of informing assessment for regulatory purposes while also looking at some different areas. So for example, common principles related to things such as protocol and report formats and promoting study transparency. So again, I think they're both complementary to each other. And we really do welcome comments as the reflection paper does progress through the process, and should a topic be proposed, please keep a lookout for that. So thank you.
Great, thank you for that answer, Melissa. Jill, back to you for the next presenter.
Terrific, thank you, Nick. And hold for 1 second, our live audience. So I'm going to ask a few questions on Q2R2 and Q14 for Dr. Keire. Dr. Keire, did the Q2, Q14 EWG consider the changes being proposed in the M4Q Common Technical Guideline guidance? Excuse me.
Thank you for the question. Yeah, we actually worked really closely with the folks that were working on M4Q, which is, of course, kind of early in the process compared to where we were. So we're kind of giving them input on elements that we were working on. So there was a lot of discussion, and ultimately we decided, because there's uncertainty in actually what M4Q is going to contain, that we wanted our guidelines that were going to be implemented much sooner to be functional guidelines current in the current state. So we included elements of the the currently enforced M4Q CTD guidelines in our submission section, but certainly those will need to be updated when M4Q is modified. Thank you for the question.
Thanks very much, David. One of our participants asked, as demonstrated in your slide by the dotted line, currently you have to flip between ICH-Q2 and 14, yet we are promoting the life cycle approach. Keeping ICH-Q2 as a separate document, unfortunately, still gives the perception of validation being a separate activity, when in fact it is integrated with development and continued verification of the true analytical procedure lifecycle. What was the reason behind separating Q2, R2, and Q14? And are there plans to combine the documents?
Yeah, thank you for the question. And I guess I'll need to take a minute here to kind of take it apart. I mean, I think an analytical procedure has a life cycle. I don't think we're promoting that or otherwise speaking to an analytical life cycle. And so, but I also would say that the EWG starting back in 2018 in Charlotte. I still remember this conversation about whether we should have one document or two. And a lot of the discussion, I would agree with the question that, what is developed is validated, right? And but then when you think about how these documents, these guidelines are used, I mean, some folks may not want to use the tools that are available and the kind of enhanced approaches that are described in Q14, but they all will use, Q2 elements, right? And so if we combine them into one document, those key validation elements are diluted in this other document, and so ultimately it was decided to keep two separate guidelines. But that was after a lot of discussion. I would say that discussion went on for 3 years and involved a lot of the constituent review. So it wasn't done idly, I guess. It was, and I think that, but our focus was on the the end use of the guidelines. Some will just use Q2 R2, while others will take advantage of what's presented in Q14. So thank you for the excellent question.
Great. Our next question is, when does FDA expect to implement Q2, R2, and Q14?
Well, hopefully in the next few months. So we've been going through all the steps, and since November to get to the implementation phase. So look for it soon. Hopefully it will be appreciated when it's available. Thank you.
Great. Thanks, David. The next question is, what are the benefits of using an enhanced approach to analytical procedure development for applicants?
Right. So there's a couple parts of this. One is, I think a lot of the firms do in development, they learn a lot about their analytical procedures, and then they kind of put it on the shelf, do a validation and submit that to a regulatory agency. And really, sometimes, as I mentioned in the presentation, sometimes the regulators don't fully understand, maybe something about a particular non-conventional technology, and so that leads to a lot of questions and delay in time. And so the idea is that we can avoid that by front-loading this work, right? And so you would apply an enhanced approach to your in analytical procedure development, develop a lot of procedure information. Maybe you're already doing that kind of thing, and this kind of gives you a tool to present it to the regulator and say, hey, look, I really have this under control in this operation space. I know that it's going to give me good results, and the regulator could say, I agree with you, that you can move around in that space without really having to do another filing, right? And so that's kind of the idea. That kind of pulls in Q12 tools, but again, ultimately, the enhanced approach gives the applicant a lot more procedure knowledge that can avoid problems, right? So even if you don't do anything and submit it to the regulator, it's still going to help you. That's my point of view. So thank you.
Great. The last question for you, David, before we move to the next presenter is, how do the Q2, R2, and Q14 guidelines relate to the guideline on continuous manufacturing?
Yeah, good question. Thank you for that. So in continuous manufacturing, there's a need to carefully control a process, and that is done by using process analytical technology tools. Often they're multivariate spectroscopy tools where you're collecting the entire spectrum rather than an isolated wavelength, and you're using a model to provide real-time feedback to a process to keep it under control and produce drugs that are the same quality over that continuous process. And so it's the kind of things that we provide in both Q2 and Q14, I think, are going to be tremendously useful to folks that are working in that space. And because we've expanded the descriptions to account for those kinds of that application. That was a real target in our discussions. And so I think it's really going to be very helpful to folks working in the continuous manufacturing space. Thank you.
Terrific. Thank you, David. Nick, back to you.
Great, thank you, Jill and David. So the next set of questions is for our Q5A-R2, which is viral safety evaluation of biotechnology products derived from cell lines of human or animal origin. So our presenter here was Christopher Storbeck from Health Canada. So Chris, our first question is, if head-to-head testing is not required for NGS validation, what is recommended for NGS validation when used for cell bank testing?
Hi, Nick. Thank you for the question. So for validation, excuse me, guidance is provided regarding validation of the method, and performing matrix-specific validation when NGS is used as a replacement method is recommended, and qualification of the method in matrix-specific verification if NGS is used as a supplementary method. So it's also clearly stated in the guidance and the revision that the validation and qualification should consider the principles of ICH Q2. Thanks.
Thanks for that response, Chris. So the next question is, are cell therapies considered to be within the scope of the ICH Q5A revision?
So thank you for the question. Cell therapies are actually excluded from the scope of the revision. However, it is stated in the revision that elements of the guidance, such as biological starting or raw material safety considerations, can be taken into consideration. So, of course, cell therapies are not a therapy that's amenable to virus clearance, and thus the reason for its exclusion. But that sort of brings into prominence the other two pillars of virus safety, and they take on a more prominent place in the overall virus safety considerations. So additionally, the virus is utilized in the generation of some cell therapies, such as CAR T cell therapies, may fall within scope, but that depends on how amenable those viruses are to virus clearance. Thank you.
Great, thank you, Chris. Next question. So whether ICH Q5A-R2 is applicable for the drugs that are produced using animals' milk source and shell and beeswax source materials as bulk raw material. Please clarify if this guideline is applicable for the above said animal source materials, non-animal source material that is non-living in nature, not like a cell or biological process-based material.
Well, thank you for the question. So ICH Q5A deals with in terms of the scope, deals with biotherapeutics manufactured from cell lines. So those types of starting materials would fall outside of the scope. However, that's not to say that some of the concepts of ICH Q5A, including raw material sourcing and testing, could not be applied to the viral safety considerations for those types of raw or starting materials. Thank you.
Great. Another question. Next generation sequencing is mentioned in the revision as a method that can be used to replace the in vivo virus detection methods and supplement or replace the in vitro virus detection method. Why does the revised guidance promote the use of NGS over the in vivo methods and in vitro method?
Yeah, excellent question. It's perhaps not so much that it's promoted necessarily, but it's based on the scientific advances and the science behind it, next generation sequencing, and essentially it comes down to the sensitivity of the methods. NGS is a highly sensitive method capable of detecting very low levels of sequences within a mixture. And considering the other methods for detection of viruses, like the in vivo and the in vitro method, aside from reducing the use of animals, the in vivo methodologies, there can be issues with sensitivity, and with the in vitro methodologies, often there are situations where the same cell line may not be able to detect all family members or all members of a family of viruses. So there are definitely issues with detectability and sensitivity with the in vitro and in vivo methods. So that is the capabilities and sensitivities of NGS allow a very broad general detection capability. And so, of course, if sequences are detected, it has to be confirmed whether these sequences are accompanied by infectious virus. And so it's really a first signal. And in that regard, it is a more stringent form of screening in a sense. Thank you.
Great, thank you, Chris. So I think we can pass it back over to Jill for the next set of questions for the next presenter.
Great, thanks very much, Nick. So the next set of questions is for Steve Mahoney on Q9R1. Steve, how does Q9R1 help industry and regulators better manage the dynamic risks that occur during the product and facility life cycles? Why don't we move on? Nick, why don't we move on and come back to Steve? Should we move on to our next presenter?
Sure, we can definitely do that. Might be some
Terrific,
technical difficulties here. Yeah, no problem. So the next set of questions will be for the presentation on cell and gene therapies discussion group, and this will be for Kathleen Francissen. So first question for Kathy, we noticed that you use the term ATMP during your talk. Is ATMP used interchangeably with CGT product?
Yeah, thanks for the question. It's really a good one. And in fact, what we see is that there are many different terms used to describe this class of products around the world. So the discussion group did really look at this more closely. And as I mentioned in my talk, we've been asked to work closely with the World Health Organization. We do have a WHO rep on the discussion group. So we looked at this question of terminology. And actually, the WHO went through quite an extensive and thorough process of looking at how you delineate these products, be they cell and gene therapies or advanced therapy medicinal product. Those two terms do tend to be used interchangeably. But the WHO did go through and engage global stakeholders in a very transparent manner. And the result of their thorough work culminated in the Annex 3 paper on human cells and tissues and ATMPs, and that was adopted in 2023. And so that's actually a very good resource where you can just see what's considered in scope and what's not, and what is some broadly recognized terminology. And so we actually agreed that ATMP really is the appropriate term to use, with it being already internationally recognized. Thank you.
Great, thank you for that, Kathy. Next question. Are there ICH expert working groups or new topics, I guess that would be, that you would anticipate the cell and gene therapy discussion group to advise of this year?
Yeah, thanks. That's another great question. As I mentioned, we have been asked by the ICH Q1, Q5C expert working group to provide input, and we've done that. We know that the ICH Q6 expert group is getting started this year, working on specifications, and we anticipate that they may reach out to us. Also, ICH M4Q on the Common Technical Document, they are quite active right now as well. And we can imagine that they might also contact us. In fact, with cell and gene therapy products, we do have some very new technologies, new types of manufacturing processes, new types of information. And so we can imagine that we would have some pretty unusual dossier structures that would work the best. So that's another one we anticipate. Thanks.
Great, thank you for that, Kathy. Next question, what are some of the major challenges that you have experienced or foresee in completing the remit of this DG?
Yeah, it's an excellent question. It's actually quite a tall order for us to assess all of the ICH guidelines for their applicability to cell and gene therapy products or ATMPs. Actually, this field is more interdisciplinary in many regards. There are many examples where clinical and quality colleagues need to work together closely. So it's actually quite good that we have clinical, non-clinical, and quality experts working on this together. This being said, it's nevertheless quite a large undertaking. We were, I would say, reasonably impressed when we pulled together all the documents that we could and found ourselves with a little over 400 guidelines available today. So we'll be going through to filter those and look to see where there's already some existing harmonization or where there are gaps. So that just showed us that it's going to be valuable to do these initial kind of exercises of level setting before we dive fully into all the ICH guidelines. Thank you for the question.
Great, Kathy. And another question, you may have already answered this, but how large is the DG and how is the DG able to handle consensus on global regulatory differences for cell and gene therapy products?
Yeah, it's actually, this discussion group is quite large. We have 48 members. We have regulators from all around the globe, all different regions. As I mentioned, we also do have the WHO and the IPRP represented here. So consensus really requires some thoughtful discussions, sharing information and documents that are currently in place. And sometimes we find that the intent might actually be aligned fairly well, even though they might be explained using different terms. So this will require some really thoughtful work for us to reach consensus so broadly. Thank you.
All right, thank you very much for those responses, Kathy. So we are going to try to go back to Q9R1 with Steve Mahoney. So let's give this another shot. Back to you, Jill.
Terrific. Thank you very much, Nick. And hold for 1 second. So Steve, how does Q9R1 help industry and regulators better manage the dynamic risks that occur during the product and facility life cycles? So it sounds like we may still have some technical difficulties. So maybe we should go to round 2. And hopefully we can work through these relatively quickly. So sorry about that.
All right, thank you, Jill. So we're actually going to cycle right back to you to go through another quick round of questions for the presenters. So next question is, have there been efforts to improve the M10 bioanalytical guidance as it applies for biomarkers? We have used the endogenous analytes section to adapt our procedures, but in most or all cases, we cannot follow guidance because we don't have a true blank matrix.
So that's a great question. ICH M10 is intended for the method validation of assays for small molecule drugs and biologics. Biomarkers are specifically out of scope. If you wish to follow the principles of M10 for your biomarker method validation, not all aspects of the guidance will address biomarker method validation, and you should provide justifications for your choices in addressing areas that differ from small molecule, drug, and biologic method validation. Great question. Back to you, Nick. Thank you.
Great, thanks. So I think that concludes the questions we got for you, Jill. So we can actually pass it right back to you for the next set of questions for M12.
So thank you very much, Nick. And it looks like we don't have further questions at the moment on M12. So perhaps we can move to our next presenter.
Yeah, we can move to E2D. We do have one more question for E2DR1 for Craig. So the question is, the definition of D0 for ICSR coming from the literature is different from the position of Inspector of Europe. Do you plan to align the position?
Yeah, thanks for the question. What I can say is that the definition of day zero and exactly how we characterize that was considered very carefully for all the sections of the guideline where we provided a time frame for reporting, but especially for the literature section, because we had a lot of discussion about that. So it's absolutely intentional where we landed and the recommendation that we're providing, and all parties, including my colleagues with the European Commission and the EMA, are well aware and understand this definition of day zero and are fully aligned with it. I can't speak to European inspectors or what may or may not need to happen for alignment on their side, but I can tell you it's very intentional, and every group on the EWG is in agreement with it. Thank you.
Great, thank you for that, Craig. So Jill, I think we're passing it right back to you. We're going to give Stephen here another shot with Q9.
Terrific. Thank you very much, Nick. And I'm just going to go back to our list of questions on Q9R1. Bear with me for just 1 second. So Steve, how does Q9R1 help industry and regulators better manage the dynamic risks that occur during the product and facility life cycles? Okay. We're still trying, everyone, but it looks like we're still experiencing some technical difficulties with Steve's mic. So let's go back to E6R3 and some more questions for Keire. Keire, one of our participants asked if you could provide a little bit more information about plans for Annex 2.
Yes, absolutely. As all of you are aware now, Annex 2 touches on designs such as the use of decentralized elements, pragmatic elements, and the use of real-world data, specifically in the context of interventional clinical trials. The outline of the guideline is very much in advanced shape at this point. Actually, we started what we call caucus review, which was we send it to experts within each organization. We included also academic experts in this review. So, and we start getting back their input at this point. And I'm happy to report that so far we've been getting very positive reviews of the guideline of Annex 2 specifically. And the group is working on it diligently to incorporate the input that we've received. The guideline itself is high level, and as Carol earlier in her presentation mentioned, it doesn't necessarily add new requirements, but clarifies how GCB concepts can be applied to those designs. We still anticipate to be on time, which means public consultations in the second half of this year. So hopefully everybody will take a look at it and provide us with input. We're excited to get interest similar to what we had in Annex 1.
Great, thank you, Keire. Next question is, what does E6R3 have to say about patient engagement?
Yeah, it's a great question. We've discussed that quite a bit because in E6 we are trying to cover the spectrum of clinical trials, all the way from early phase kind of proof of concept when you have very few, sometimes it can be counted in one hand, number of participants, to the more complex, phase 3s, large clinical trials, as you can imagine. So, for early phase, certain level of engagement may not be needed or may not make sense when you have just a proof of concept study in a few individuals, for example. However, not just participants' engagement, but also community engagement may be very relevant at large-scale clinical trials. So the current language encourages engagement with the stakeholders, and they include patients and participants in that, but as well as healthcare providers. For example, if you want to work with community healthcare providers, you might want to consider also engagement with them at earlier stage. And just to be very transparent with everybody, in a lot of the comments we received asking us to bolster a little bit the language we have around community and patient engagement, and that's something the EWG is working on at this point.
That's great. Thanks very much, Keire. Nick, why don't we move on to the next presenter?
Sure, thanks, Jill. Just working through questions here. So yes, the next presenter will be back to David on Q2 or Q14. So the question is, how important is it to include a criticality score accounting for impact and uncertainty in risk assessment for analytical methods? And how can this criticality score be integrated into the risk assessment ranking system effectively?
Well, thank you for the question. I think Steve would probably opine on this. Definitely, I'd love to hear actually his answer to this question. Unfortunately, we're having those technical difficulties. But I say that's this criticality accounting is a tool that can be used. We talk about risk assessment in Q14, but we're not advocating the use of one tool over another. That it's good that you're doing it, and that's one approach. And the second part of this question, can it be done effectively? Right. That's kind of an implementation question, and maybe more appropriate for Steve Mahoney to answer than for me. But I think it's a tool, but it's not the only approach that you can take to do these risk assessments. It's certainly one that a lot of people do use, and there's a host of scientific literature describing effective ways of doing that. Thank you.
Great, thanks very much, David. So I think that concludes the questions for Q2 or Q14. So Jill, back to you.
Great, Nick. So we can move on to some additional questions for Chris Storbeck and Q5R2. Chris, you mentioned that for certain well-characterized cell substrates, in vivo testing may not be necessary during cell line qualification. Could you expand on this a bit?
Yeah, thank you for the question. Certainly, and this pertains to a situation where the sponsor has extensive experience with that particular cell line. It's a well-characterized cell line, such as a CHO cell line, for example. So if they've performed in vivo testing on the parental cell line, and they've generated several master cell banks for other similar products by a well-controlled process, then in that situation, the in vivo testing requirement can be waived. Thank you.
Great. The next question is, could you please give a few examples of the use of prior knowledge to achieve regulatory relief? Yeah,
certainly. So prior knowledge, and certainly the answer I just gave is one example of the use of prior knowledge. There's other examples as well. For example, with resin reuse, as I mentioned, with the protein A chromatography steps, and the also the anion exchange and cation exchange chromatography steps, where in certain situations prior knowledge can be used regarding reuse of those columns. So, and that comes down to a knowledge of the process, knowledge of the process intermediates. Also for leveraging virus clearance studies for a new product, using data from a well-characterized platform process can be acceptable. So for certain virus clearance steps, such as low pH, detergent solvent inactivation, and virus filtration. In the case of virus filtration, one product-specific demonstration of clearance has to be submitted. That's a recommendation in the guidance. But certainly where the process parameters are well understood, the process intermediates and the composition of those intermediates are understood, then those that information can be used to gain some regulatory relief. But it has to be laid out and scientifically based during the submission. Thank you.
Thanks very much, Chris. The next question is, there were many for your section for Q5A. Suppose a research protocol proposes using a novel biotechnology product derived from cell lines predominantly representing a single ethnic group. The product is intended for a disease that disproportionately affects a different population. Dr. Storbeck, how might the Q5R2 guidance inform an IBC's review of a protocol like the one described? Are there specific concerns the IBC should raise regarding the potential impact of source cell line diversity on the product's safety and efficacy across the intended patient population?
Well, thank you for the question. Yeah, it is. So it is. ICH Q5A does address the manufacture of biotechnological products derived from cell lines, and I think it's more important to consider the species of cell lines with respect to safety, efficacy, and risk, rather than specific ethnic groups within a species. So through the lens of scientific understanding and genetics, and there's certainly going to be some genetic differences, but overall, from a species perspective, where the similarity is to such an extent that the granularity of different considerations between ethnic groups, I don't think is possible. But thank you for the question.
Thanks very much, Chris. Okay, Nick, I'm going to send it back to you.
Great, thank you.
Yeah, sure.
Yeah, sorry. I think we've got a few more minutes left, maybe a chance for another couple of questions. But thank you very much, Jill and Chris, for that. So I'm going to go back to the Cell and Gene Therapies discussion group, Kathy. So the next question is, what are your thoughts on the advancement of the therapeutic products under the initial scope of the discussion group, and how the remit will help in advancement of these products and other cell and gene therapies products, especially in light of the fast speed of technological development of ATMPs?
Yeah, thanks so much for the question. Actually, we continue to see quite a number of AAV-based gene therapy products and CAR T-cell products going to market, and they're going more globally. And so that experience helps everyone see where they've really matured and what the requirements are to make it through development and on to approval. The other thing it does is it highlights the current state of where these products are. And so it's helping us think about what are the requirements currently, but what do we think will be required in the future? And that should really help set the stage for anticipating a lot of emerging technologies that are coming along in this field. They do, in fact, come along very quickly. So our working through these initial product classes will be quite helpful. Thank you.
Great, thanks, Kathy. And I think we've got time for one more. So another one for you, Kathy. The discussion group is for cell and gene therapy, but the presentation refers to the term ATMP. Can you please explain the significance of the use of the term ATMP versus cell and gene therapy?
Yeah, thanks, Nick. I think we kind of had that question earlier in it, and you can use either of these terms interchangeably. But indeed, as we looked across for really the best resource, we turned to the WHO and the terminology that they put forward in a very thorough way. It's an excellent resource, and so we do feel that using the term ATMP is quite appropriate. Thank you.
Great, thanks very much, Kathy, for those responses. So just looking towards our coordinators here, or Jill, or do we have time to take any other questions, or are we looking to close off?
I think we're right at time, Nick. Our participants have had great questions. So
great, yeah, no, definitely a lot of great questions, a lot of great information in today's session. Unfortunately, never enough time to get through everything, but we wanted to thank everyone for participating today. We hope you found it very informative. Thank all the presenters as well for their time in providing this very valuable information, and wish you all a great day. Thank you very much.
Thanks so much. We do have a few housekeeping reminders. Please remember the 14-day deadline. That's March the 7th to claim the certificate of attendance to support CE for SOCA, RAPS, SQA, and ACRP. Remember to download the certificate immediately upon creating it. Otherwise, this opportunity will be lost. Recordings will be posted in approximately 7 to 10 business days. On behalf of CDER Small Business and Industry Assistance, we hope that you enjoyed today's presentations and found them helpful. We look forward to your presence at our next SBIA webinar. Thank you for attending, and this concludes today's webinar.